Hello friends, in this video I am going to tell you what are the questions that you can challenge and what are the questions that you cannot able to challenge regarding to shift to question paper. So first question is question number 240. The following statement was made regarding the patterning of anterior posterior body plane of Drosophila. Okay, and the correct answer that is from NT they just mentioned option number 950, that is option number A and option number C are correct like that they just mentioned. Okay, so this option is absolutely correct. In the developmental biology Gilbert book itself they just mentioned when you inject a bicoid mRNA in the middle of the bicoid deficient embryo, definitely it will be leading to the formation of head in the middle and tesla at the end of two cells. This option is absolutely correct. Next thing, nanose protein inhibit the translation of caudal mRNA at the posterior half of embryo. But please understand friends, only bicord protein will be inhibiting the translation of the caudal mRNA at the anterior post, whereas this nano protein will be inhibiting the translation of hunchback mRNA at the posterior half of the embryo. So this option is wrong only friends. Okay. So next, bicoid protein activates the zygotic expression of hunchback. These are correct. And the last option is I am still doubting only this last option friends, because option number A, C, D, that is this option number 959 should be correct, because segment polarity gene are expressed in the segments of embryo. That's a correct option, even though I didn't go able to get any proof for this particular thing, but option number D is also correct. So option number A, C, D is a correct option. So you can claim you can challenge this question. Okay, so the next question is question number 241. Okay, so here they just mention the group of cells called vulval precursor cells of the cell ligand form a equivalence group. The following statement were made as evidence of VPC's form a equivalence group. So for this question they just says 962 is the right option, like option number B and option number C is correct like they just mentioned, but option number A is also correct friends, like A, B, C is the correct option. Okay, so this option, that is A, B, C is a correct option, because if you destroy the anchor cells at the vulval precursor cell, definitely they will be contributing the formation of hypodermal cells. That's the correct option. Even you can find this in a Gilbert book also. So let me show you. So here you can be able to see, this is from page number 99 in Gilbert 9th edition friends. If the anchor cell is destroyed, the VPC, that is vulval precursor cell, will not form vulva, but instead it will become a part of hypodermis or hypodermal cell. Okay, so option number A, B, C is a correct option. Okay, so you can quote the Gilbert page number itself, they will be 3. Okay, option number A, B, C are correct. Okay, next question is option number 185. Option number 185, a type of regeneration in which a differentiator cell divide maintaining the differentiated function without de-differentiation and production of undifferentiated mass, and they are saying compensatory regeneration is the correct option. Of course it is a correct option, but the question is wrong friends. Okay, so let me tell you about this particular article, or it is taken from Gilbert book Developmental Biology only friends. So here what it just mentioned means, so here the third type of regeneration is an intermediate type and can be thought as the CR, that is compensatory regeneration. The cells will be dividing, but at the same time they will be maintaining a differentiated function. They will be producing a cell similar to themselves, but they do not form a mass of undifferentiated tissue. But what is given in the question? They will be producing a mass of undifferentiated mass. So this particular question is wrong or confusing. Why I am saying confusing means, if the answer, if the particular dialogue is continuous, like if they are saying a type of regeneration if the differentiated cells divide maintaining the differentiated function without de-differentiation and without the production of undifferentiated mass. If they had given clearly means that fine, but here they are saying without de-differentiation and production of undifferentiated mass. But when I check the Gilbert book, they are saying they will be producing, that is, so here I just mentioned you, so they do not form a mass of undifferentiated tissue, but in the question they will be producing a undifferentiated tissue. So it is a confusing question, so you can challenge and saying that question itself asked wrong. Okay, and next thing is regarding to question number 226. So question number 226, it's so easy. I think many will be attempting this question so that you will be getting grace mark. If you check the option, that is this option, okay, option number A and option number D, so both the options are similar only friends. If you check finally you can able to see both the options are same only. Okay, but here they had demanded option number A and option number B are correct, but option number D is similar option. I think there is a typing error. That's why they have typed the same option in D. They should be asked some other answers. Okay, so anyway, this question will be considered as a dropped questions, and student who had attempted this question, you will be getting your marks. Okay, because the option, that is option number A and option number D are similar or same. That is due to any mistake they have typed this particular question. And next question is question number 230. So here they just says some steps in process of eukaryotic DNA replication mentioned below require hydrolysis of ATP. So and the options are, that is 920, that is B and D. They just say that DNA strand separation by helicase and joining of Okazaki fragment alone require hydrolysis of ATP. But I am challenging this question, that is camp loader associated with the clamp and DNA also requires the hydrolysis of ATP. That is, if you check this particular article, so here they just mention the assembly of a clamp around the DNA requires the ATP hydrolysis by a special protein complex, the clamp loader, which will be hydrolyzed the ATP as it loads the clamp on the primer template junction. So you can challenge this particular question. So it is available in the molecular biology. So in that they had clearly mentioned you about, friends. So for this particular option, option number B, option number C and option number D, that is this particular option is correct. Okay. So for those who have attempted a question, definitely you will be getting a mark. And the next question is question number 238. Question number 238. Okay, so there is nothing wrong in this particular question, friends. So please don't challenge this question. You can refer QBA book, friends. So that QBA book here they just mentioned under the figure 7.2. So in the QBA book, I think it's page number 164. Here they just mentioned the progeny of the crosses between two inbred strains with a different MHC haplotype will be expressing both the haplotype. At the same time, they will be accepting a graft either from both the parent, that is either from either of the parent as well as from one another. So if you check the answer, this is that skin graft from any parents will be accepted by the progeny of recipient. It is the correct option. So please don't challenge this question. I had analyzed in a QBA book. You can't able to challenge this particular question. Next question is regarding to question number 260. That is with regarding to maternal effect gene. Okay, so here what they say is that the particular cross 5 only is wrong, like that they were saying about during a development many gene products are provided by the female to egg which are needed for the normal development of zygote. Such genes are called as maternal effect gene. The following set of crosses has been made between a parent carrying a recessive mutant allele and the offspring that is after. And they are asking which of the above crosses is an indicative that a mutation is a maternal effect gene, like that they are asking. Okay. Again, this question will be considered as a dropped question because you can able to see here, that is cross number 1, the genotype of parent, and if you check the cross number 5, that is a genotype of parent, both are similar. And in this cross, the cross number 1, they said that all phenotype will be normal, like that they just mentioned. Whereas in cross number 5, that is the same genotype, here they are saying both the normal and mutant will be arising. So the question and this particular tabulated column that they had derived itself wrong. Okay, so you can arise a question wrong issue in case if they are providing the thing, means definitely we will be getting a mark. Okay, the next question to challenge means it is question number 174. So this is what thing, which one of the following statement about the DNA replication is incorrect? And for this question they says that option number A, that is once the DNA replication commences, it always continues uninterrupted until the entire process is incomplete. If you simply know about the process of leading strand synthesis and lagging strand synthesis, then you can correctly attempting this question. This is an incorrect statement. Whereas if you take the eukaryotic genome replicate from multiple origin of replication, absolutely a correct statement. Okay, there are numerous multiple origin of replications will be available in eukaryotic genome as well as the consensus sequence, that is autonomously replicating sequence, that is ARS sequence, that has been originated from E. Saccharomyces cerevisiae. And the last option is also absolutely correct. The fully methylated DNA sequence as well as non-methylated, this origin of replication C can initiate the DNA replication, whereas the semi-methylated DNA cannot able to initiate the DNA replication, friends. So you can check this particular article. So here they just mention the hemi-methylated Hemimethylation prevents the DNA replication in E. coli. So please don't challenge this question. Anyway, if you challenge, they won't accept your claim. Okay. So the next question is with regarding to question number 273. Okay. So in this question, here this is mentioned many. I don't know how students are attempting these questions. Okay. So many students. So in for this particular question, is question number. Option number A and C are correct, friends. Okay, so they are asking incorrect statement. So option number A and option number C are correct. Based on my knowledge, the answer that is given by NT is absolutely correct. That is only this particular option, that is sampling mode plot will add more species in site B but not in site A is correct. And option number D, that is site B is likely to have high species distribution than site A is correct. But they are asking what is the incorrect statement. And option number A, that is in both sites sampling mode plot will not add any more species. That's the incorrect species. It will be adding more species. Okay. Next thing is regarding to option number C, that is site A and B are likely to have similar species distribution. That's the incorrect species. Even though the sites are being situated, the species distribution will be different. Next question is option number 255. So option number 255, yes, many students are asking this doubt, but entire thing is correctly matched, friends. The ferritin is a protein that is playing an important role in intramucosal cell ion binding. And with regarding to ferroprotein, yes, this particular ferroprotein is involving in process called when an ion leaves a mucosal cell through this ferroprotein only. Next, transferring, you all know, plasma, it is a plasma ion binding protein, and hepacitine, it is so hypoxia is known to reduce the synthesis of hepacitine. Okay, so I'll be giving you a definition of this particular hepacitine. So you can find this particular article. So here they just clearly mention you, that is hypoxia is another suppressor for a hepatic hepcidin protein expression independent of body iron level. Okay, so you can refer this particular article. So please don't challenge this question. The option and the match, they have correctly matched, friends. So hepacitine, so hypoxia is a suppressor, that's why this hypoxia will be reducing the synthesis of the hepacitine. So the next question of choice is with regarding to question number 188, friends. Question number 188. So this particular question again, identify the correct action site of action of this DBMIB, that is 2,5 dibromo 3 methyl 6 isopropyl benzoquinone. So I think you might be know about this DCMU. This particular thing is a different, and it is a correct option only, friends. That is option number 751. So if you refer this particular thing in this article, they just clearly mention you about the action of this particular photosynthetic inhibitor. So it is a plastoquinone analog which will be binding with the cytochrome B6F, that is cytochrome B6F, and it will be preventing the transferring of electron from a reduced plastoquinone to cytochrome B6F. Okay. So the correct option, that is 751. Okay. So please don't challenge this question, friends. Next question is with regarding to question number 231. So here you can able to see, I don't know how you are attempting these questions on all, friends. It is even though it is easy, but it's really hard, and it really takes around 10 minutes time point to solve this question. Anyway, I won't take this kind of questions and all when I was writing the CSIR examination. Here they just mention option number C. Okay, so that is the 921. Option number C is alone correct, like they're just saying. Insertion of A after the position of 26 changes the primary structure of protein and results in a truncated protein synthesis. So if you take the position number 26, 1, 2, 3, 4, 5, 6. Okay, so if you change that particular thing, that is insertion of A after the position of after the position of 26, this alone is a correct thing. Whereas option number A, option number B, and option number D are incorrect. Okay, so deletion of A at a position of 9 creates a stop codon. So in order to solve this particular thing, if you know about the stop codons, you can able to sense, and both the option number A and option number B are incorrect only. Here they just mention when you substitute time at position number 2, it will be changing the primary structure without altering the transcript length. The entire statement itself wrong. The same time, when you insert a nucleotide at position of 8, it will be increasing the nucleotide transcript by 1 and will be changing the amino acid sequence of protein being translated. It is also a wrong option because there is a process called, there is an important concept called genetic code redundancy. Okay, so there is nothing wrong in that. Next question is with regarding to question number 271. So question number 271 here. There was a question regarding to monitoring protocol, and the correct option is 1 or 2. That is, they says that only option number B and option number C are correctly they just mentioned, because polar vortex is not only formed by north pole, it is also formed by south pole. Okay, at the same time, more extensive formation of polar stratosphere cloud over South Pole compared to North Pole. Again, this is a wrong option, friends. So this is a correct option. This particular option number C. So from ND they just mention option number B and D are correct option only. So it is correct option. Please don't challenge this question. An emission of ozone depleting substance are higher in southern hemisphere compared to northern hemisphere, like that they are mentioning. So this is a wrong option. That's why the option they didn't mention, because with regarding to emission ozone depleting substance, they are higher in northern hemisphere when you compare with the southern hemisphere. Okay. So the next question is regarding to question number 258. So 258 is with regarding to this particular question. So please don't challenge this question. It is absolutely correct, and try not to challenge about this kind of application oriented concept, because you can't be able to collect a sufficient evidence, and the same time from India, from CSI, they won't easily accept a challenge at all. Okay. So next question is question number 179. So 179. Again, this particular they are asking which of the following statement is incorrect, and option number C, that is incorrect. They are asking the function of, that is option number C, that is a function of P53 and RB are augmented by E6 and E7 protein of human papilloma virus. Okay, they are asking which of the option of incorrect, but this option is absolutely correct, friends. Okay, let me share with you about this particular thing. Okay, so if you check on this particular article, they had clearly mentioned about the E6 protein and the E7 protein of the human papilloma virus, friends. Okay, so in that they had clearly mentioning. So in this article they had clearly mentioning you, friends, that is E6 and E7 contribute to the achievement of the uncontrolled proliferation to degradation of growth suppressor. So E6 they have a target on important growth suppressor, that is P53, while this RB, retinoblastoma protein, is a major target of E7. What is asked in the question? So P53 is again augmented by E6 and RB is again augmented by E7, but I think here P should be coming, RB protein should be coming. So that's why they are asking incorrect statement like that they are asking, because it's not simply RB, the P will be coming. So if you check on this particular thing here, you can able to see protein RB, that is a retinoblastoma protein. So that's why it's a spelling mistakes. I don't know, better if you want you can challenge this question, friends. Okay, so that's why from NT they are asking incorrect statement, but the function is augmented is absolutely correct. Here P is missing. It might be new to typing error, whatever it might be. Okay, so if you want you can challenge that question, but absolutely it is a correct statement. Okay, so the next thing is regarding to question number 234. So 234, it is also a correct option, friends. Okay, so please don't challenge this question. I just refer in QB book also. Next is question number 165. So 165 question. So this is also a correct option. If you want check, some will be solved in part A section, so you can get a reference from them. And next question is regarding 265 question. 265 question, which of the following statement is not correct, like that they are asking. So they says that option number D, that is 1060, all corals grow only in photic zones as they need a sunlight to grow. This is absolutely a wrong statement and incorrect statement, because the deep sea corals will not, they don't require sunlight for their growth because they are in deep sea, whereas only the shallow water corals alone will be requiring sunlight. Whereas corals processing a special cells called nematocytes, it's absolutely correct option, and several corals will be having a mutualistic interaction with the zooanthid, it's also correct option, and reefs form when coral grow in a shallow water closer. That's why they are forming coral reefs. Okay, so all the three options are correct only, but they are asking which of the following statement is not correct, that is incorrect. Okay, so this option is an incorrect statement, so don't challenge this question. Next question is question number 274. Okay, so I don't have any much evidences to challenge this particular question. If you have any evidences, then please do tell me about them. Okay, so here you can able to see, I think this question will be considered as a dropped question, friends. Okay, many questions that is asked in shift 2 they are considered as a drop, because here in option they are saying both option, like here tropical rainforest and tropical rainforest, like that they mentioned, cultivated land and open ocean and open ocean. So I think they had done any typing error, or it might be due to any technical error. I think the option itself given wrong. Okay, so I think this question will be considered as a dropped question. Next question is question number 176. Okay, so this is with regarding the post-translation modification, one of the core histones that are known to be achieved with DNA repair pathway. The correct option is ubiquitination of specific lysine residues, whereas if you take phosphorylation, phosphorylation does not occur in tyrosine residues, it will be occurring serine residues. Whereas if you take the acetylation, here as they says that acetylation has a serine residue, but generally acetylation will be done in a lysine and histone 3. Okay, and methylation, generally methylation will not be done in serine residues. Methylated will be done in only in arginine amino acid and lysine amino acid. So the correct option is ubiquitination at a specific lysine residues. If you want in-depth analysis of this, I will be adding few paper for you, so you can refer this particular paper. So in this paper they just mention you clearly about the post-translation modification in DNA repair pathway. Okay, and next thing is with regarding to question number 270. Question number 270. So again these things and all don't challenge, because I just found a research article. In that article they just mention, so here you can able to check. In this particular article they just mention fragment size and connectivity were related to the species richness. Okay, so what did they ask? Species richness will depend upon a fragment size. Absolutely correct statement. And species richness will depend upon the physical connectivity between fragments. So what the option that is given by NTA, it is correct. Don't challenge this option. So the last thing is with regarding to question number 239. Okay, so 239 they just mentioned for 239 956, that is A, B, C are correct option. This is a correct option, friends. Okay, so you can check the Gilbert book. They have clearly mentioned you about this particular thing. Okay, so option number 956, that is A, B, C are correct option. Okay, all the given statements are correct only. Okay, so if you want in-depth analysis, you can also refer in this particular NCBA. This is nothing but this is a page that is taken from Gilbert book only. So here also they had given a detailed statement about that particular thing. And the next question is with regarding to question number 203. So which of the following statement is not correct? Again, here they just mentioned 811, that is gamma diversity is a sum of alpha diversity. No, gamma diversity can be expressed as a product of alpha and beta diversity. At the same time, they can be expressed as a sum of alpha and beta diversity. And both the alpha diversity and gamma diversity, they are useful for measuring the presence or abundance of species. Whereas here they just mentioned gamma diversity is a sum of alpha diversity. So this is a wrong option. Okay, so answer given by NTA itself correct. So please don't challenge this question. I hope this video will be very much helpful. If you want some other question that you need to challenge, or if you want to know whether the given option is correct or not, please type the question below. I will be analyzing and telling you. Thank you friends. Thanks for watching this video.